For more than a century, one particular grave outside the old Viking trading town of Birka, on a small island in Lake Mälaren in Sweden, sat in the archaeological record as something close to a textbook illustration. It was the grave that generations of scholars pointed to when they wanted to show students exactly what a high-status Viking warrior burial looked like: a sword, an axe, a spear, a battle knife, a quiver of armor-piercing arrows built specifically to punch through mail, 2 full shields, a complete set of gaming pieces laid, out on a board, the kind used to plan and rehearse battlefield tactics. And lying alongside the human remains at the bottom of the grave chamber, the skeletons of 2 horses, a mare and a stallion, buried in full, riding tack. It was, by any measure archaeologists used to judge these things, an elite military burial, and for well over 100 years after its excavation in 1878, nobody seriously questioned who, or more precisely, what kind of person, must have been buried inside it. Then modern genetics looked at the bones directly, and the answer that came back did not match the assumption the grave had been built on. The burial in question is catalogued in the archaeological record simply as BJ 581, one of more than 1,000 graves excavated at Birka by the Swedish archaeologist Hjalmar Stolpe, beginning in the late 19th century. Birka itself was one of the most important trading hubs of the Viking world, a fortified island settlement that flourished from the late 8th century into the early 11th, sitting at the crossroads of trade routes that connected Scandinavia to continental Europe, the British Isles, and via the river systems of Eastern Europe, all the way to the Byzantine and Islamic worlds. It was a place where wealth, status, and military power were on regular display, and BJ 581 was positioned, quite literally, right beside the settlement's garrison fort, overlooking the harbor, the kind of location that would have gone to someone of real consequence. For most of the 20th century, the identification of the BJ 581 occupant as a male warrior of high rank was treated as settled fact, repeated in textbooks, museum placards, and academic papers without serious challenge. Excavation plans drawn by Stolpe's own team in the 1880s labeled the grave in exactly those terms, and later generations of scholars, working from those plans and from the sheer weight of the weaponry inside the chamber, saw little reason to revisit the question. It was, after all, precisely the kind of grave that fit neatly into a broader cultural narrative about Viking society that had been building since the 19th century, one dominated by images of seafaring mail raiders, and largely uninterested in complicating that picture with evidence that pointed the other way. Then, toward the end of the 20th century, osteologists working through the Birka skeletal collection began noticing something that did not sit comfortably with that assumption. The pelvis was too narrow for a typical adult male. The facial bones were, by the standard morphological measures researchers used to estimate sex from a skeleton, more consistent with a female individual than a male one. Bone-based sex estimation is not infallible, particularly with skeletons that have been buried for over 1,000 years and subjected to the wear, breakage, and mineral leaching that come with that kind of age. So the finding remained a point of quiet, unresolved debate rather than an accepted revision. Skeletal morphology could raise the question. It could not, on its own, definitively answer it. For years, that left the field in an uncomfortable holding pattern. A well-known, heavily cited grave whose central identifying label, warrior, implicitly understood to mean male warrior, rested on evidence that a growing number of specialists privately doubted, but that no one had yet been able to conclusively overturn. Answering it definitively required something that 19th century archaeology had no access to at all: the DNA itself. It is worth pausing on just how difficult it is to extract usable genetic information from remains this old before turning to what that information revealed. DNA begins degrading the moment an organism dies, breaking down into progressively shorter fragments and accumulating chemical damage as decades turn into centuries. By the time a skeleton has been in the ground for 1,000 years, especially in a climate with freeze-thaw cycles and fluctuating moisture like Scandinavia's, the surviving genetic material is typically fragmentary, chemically altered, and easily overwhelmed by the DNA of the bacteria, fungi, and soil organisms that have spent centuries colonizing the bone. Researchers working on ancient human remains have learned, largely through trial and error over the past 2 decades, that certain parts of the skeleton preserve DNA far better than others. Teeth, protected by a hard enamel shell, and the dense petrous portion of the temporal bone, a pyramid-shaped structure located just behind the ear, both tend to shield genetic material from the kind of bacterial infiltration and moisture exposure that destroy DNA elsewhere in the body. It was material of exactly this kind, a tooth and a sample from the individual's upper arm bone, that the BJ 581 research team turned to when they set out to finally settle the question the osteologists had raised. Once extracted, the genetic material had to be sequenced, compared against reference databases, and checked rigorously for signs of modern contamination, since a single stray skin cell from a 19th century excavator Or a 20th century museum curator could easily corrupt a result if researchers were not careful to distinguish authentically ancient DNA with its characteristic patterns of chemical damage from more recent genetic material that might have found its way onto the bone in the century and a half since the grave was first opened. This is painstaking, unglamorous laboratory work, and it is the reason a question that had lingered since the late 1800s took until 2017 to finally be resolved with real scientific confidence. In 2017, a research team led by the archaeologist Charlotte Hedenstierna Janssen at Uppsala University set out to resolve the question using ancient DNA analysis, extracting genetic material from a tooth and from a bone in the individual's upper arm. The results, published in the American Journal of Physical Anthropology, were unambiguous in a way that skeletal morphology alone could never quite manage to be. The genome-wide sequencing data showed no meaningful presence of a Y chromosome at all. The ratio of reads mapping to the Y chromosome versus the X chromosome came out at a level the research team's statistical threshold classifies as conclusively female, with essentially none of the genetic signal that a male skeleton would be expected to produce. The individual buried in grave BJ581, interred with a full arsenal of weaponry, 2 horses, and a battle-planning game board, was a biological woman. The researchers went further than simply confirming sex. Using strontium isotope analysis on the individual's teeth, a technique that can reveal information about where a person spent their childhood based on the chemical signature absorbed into tooth enamel from local food and water, they determined that the woman in BJ581 had likely not grown up in Birka itself, but had come from somewhere in southern Scandinavia, possibly southern Sweden or Denmark, and appeared to have moved around considerably during her lifetime. Evidence of exactly the kind of long-distance mobility that characterized the broader Viking world she operated within. Mitochondrial DNA, extracted from the remains, placed her within a genetic lineage, designated haplogroup T2b, that connects to populations found across northern Europe today. Taken together, the genetic and isotopic evidence painted a picture not of an anomaly plucked from nowhere, but of a woman who had traveled, who held a position of clear military and social standing far from her place of birth, and who was buried with the full trappings that Viking society reserved for its highest status warriors. The research team's own conclusion, stated plainly in their published findings, was that grave BJ581 represented the first genomically confirmed burial of a high-ranking female Viking warrior, a formal DNA-backed answer to a question that osteology alone had only been able to gesture toward for decades. The finding did not settle quietly into the archaeological literature. It generated substantial pushback, much of it focused not on the genetic sex determination itself, which was methodologically sound and difficult to dispute on technical grounds, but on the interpretive leap from this individual was biologically female to this individual functioned as an active warrior. Critics pointed out that grave goods do not automatically tell us what social role a person actually performed in life. A sword buried alongside a body could reflect inherited status, symbolic authority, or the social position of a family, rather than direct proof that the person wielded that sword in combat. Some archaeologists also raised concerns about the original 19th century excavation records themselves, questioning whether all of the skeletal material cataloged as belonging to BJ581 could be confidently traced back to a single, undisturbed individual, given how excavation and record-keeping standards from the 1870s compare to modern archaeological practice. Some of the sharpest published criticism argued that the research team had moved too quickly from a genetic fact to a social conclusion, and that the label warrior carries assumptions about combat, training, and battlefield participation that a grave assemblage, however impressive, cannot fully prove on its own. Defenders of the original interpretation responded that the sheer completeness and specificity of the weaponry, including armor-piercing arrows designed for a very particular kind of combat, and a gaming set associated elsewhere in the archaeological record specifically with tactical planning rather than casual recreation, makes an interpretation centered on genuine military function considerably more plausible than alternative explanations resting on symbolic status alone. These are legitimate scholarly objections. The debate over exactly how to interpret the Birka warrior grave, more than half a decade after the Genomic study's publication, has not been fully resolved to the satisfaction of every researcher working in the field. But the core genetic finding, that this specific, unambiguously high-status warrior burial belonged to a woman, has held up under continued scrutiny, and it fits into a wider, independently documented pattern. BJ581 is not the only known grave in the Viking world where a person was buried with the complete equipment of a male warrior despite being biologically female. Other, less extensively studied examples have been noted by researchers working across Scandinavia, suggesting that whatever social role this represented, it was not confined to a single isolated case. Long before genetics entered the conversation, Old Norse literature had already preserved a persistent, if contested, tradition of armed women, Sagas, skaldic poetry, and later medieval chronicles describe figures often translated into English as shield maidens, women who take up arms, lead war bands, or fight alongside men in battle. For generations, historians treated these figures largely as literary invention, dramatic embellishments added by medieval saga, writers working centuries after the events they claim to describe, reflecting narrative convention rather than lived Viking Age reality. The near total absence, until recently, of any physical or genetic evidence for real female warriors made that skeptical reading easy to sustain. The BJ 581 findings did not prove that any specific saga figure was historically real, and researchers have been careful not to claim that they did. Sagas remain literary texts, shaped by storytelling conventions, oral transmission across generations, and the particular concerns of the Christian scribes who eventually wrote them down centuries after the Viking Age had ended. But the genomic confirmation of at least one high-status female warrior grave has meaningfully shifted how scholars weigh the plausibility of the broader literary tradition. The Birka grave offers a vivid, individual-level illustration of how directly ancient DNA can overturn inherited assumptions, but it is only one grave. The far larger and more sweeping picture of who the Vikings actually were, genetically, came 3 years later in 2020, when a team led by the evolutionary geneticist Eske Willerslev of the University of Copenhagen published what remains the largest ancient DNA study of the Viking world ever undertaken. The project, published in Nature after roughly 6 years of work, sequenced the genomes of 442 individuals recovered from more than 80 archaeological sites spanning Scandinavia, the British Isles, continental Europe, and Greenland, with remains ranging in age from the Bronze Age through the early modern period, and concentrated heavily on the Viking Age proper, roughly 750 to 1050 CE. What that genetic data revealed was, in Willerslev's own description, enough to meaningfully change the picture of who a Viking actually was. The popular image of the Viking, pale-skinned, light-eyed, blonde, and drawn from a genetically uniform Scandinavian stock, turned out to be substantially inaccurate even for the people living inside Scandinavia during the Viking Age itself. Perhaps the single most consequential finding to come out of the 2020 study concerned the relationship between genetic ancestry and Viking identity itself. And here, the genomic evidence dovetails directly with what the Birka grave had already hinted at in miniature. Researchers analyzing burials in the Orkney Islands, off the northern coast of Scotland, found individuals interred in full Norse style, complete with Viking swords and burial customs, whose genomes showed no meaningful Scandinavian ancestry at all. Genetically, these individuals more closely resembled the local Pictish populations native to Scotland and Ireland than they did anyone from Norway, Denmark, or Sweden. They had, in every genetic sense, no Viking ancestry whatsoever, and yet they had been buried by their own communities as Vikings. The implication researchers drew from this finding was significant. Being a Viking, in the way the term was actually lived and understood by the people of the period, was not primarily a matter of ancestry or bloodline at all. It functioned much more like a cultural identity, an occupation, or a chosen way of life, closer, as later commentary on the study put it, to a job description than a genetic inheritance. People with no Scandinavian ancestors could become Vikings by adopting Norse customs, participating in Norse trade networks, or joining Norse raiding and settlement expeditions, and their communities buried them accordingly, using the same weapons, the same rituals, and the same symbolic markers of status, regardless of what their genomes actually contained. The 2020 study also produced granular, almost novelistic detail about the internal social structure of specific Viking raiding and trading expeditions, made possible only because ancient DNA can establish direct biological kinship between individuals buried at different sites in a way that skeletal analysis or grave goods alone cannot. In one burial site in Estonia, researchers identified 4 brothers who had died on the same day and were buried together alongside a 5th individual believed to be a close relative, most likely an uncle. A genetic signature consistent with a single family unit wiped out together during a single violent event, almost certainly connected to one of the early, ill-fated Scandinavian raiding expeditions into the eastern Baltic. The study additionally found that genetic diversity within Scandinavia itself was distributed unevenly. Coastal trading hubs and island settlements, the commercial gateways of the Viking world, showed considerably higher levels of genetic mixture and outside ancestry than inland Scandinavian communities, which remained comparatively more genetically stable and homogeneous throughout the period. This pattern makes intuitive sense once stated plainly. Trade brought people and people bring genes, but that mixing concentrated heavily around the ports and markets rather than spreading evenly across the entire Scandinavian interior. But it had never before been demonstrated with this level of direct genetic evidence. Researchers involved in the project described the scale of the resulting dataset as a genuine computational challenge in its own right, requiring new, approach is simply to manage and compare genetic information drawn from hundreds of individuals spread across such a wide range of dates and locations. A follow-up study published in 2023 in the journal Cell, led by researchers including the molecular archaeologist Anders Götherström at Stockholm University, added a further, more finely resolved layer to this picture by focusing specifically on genetic change within Scandinavia across a 2,000-year span, drawing on nearly 300 ancient genomes, including new material recovered from sites such as the wreck of the 17th century Swedish warship Kronen. That research traced how ancestry from the British and Irish Isles, from the eastern Baltic region, and from southern Europe ebbed and flowed into different parts of Scandinavia at different points in time, with British and Irish ancestry showing up broadly across the region during the Viking Age, while eastern Baltic ancestry concentrated more specifically in central Sweden and on the island of Gotland. Viewed against that longer timescale, the Viking Age looks less like a sudden genetic rupture and more like a particularly intense and well-documented chapter within a pattern of contact and mixture that had already been underway for a very long time. What connects the single, vivid story of grave BJ 581 to the much larger genomic surveys that followed it is a consistent pattern. In case after case, the assumptions that had been built up over more than a century of archaeology, folklore, and popular retelling did not survive direct contact with genetic evidence. None of this means that the archaeological and historical scholarship that preceded modern ancient DNA analysis was careless or without value. Grave goods, burial customs, isotope signatures, and historical texts all continue to provide essential context that DNA alone cannot supply. DNA can tell researchers who someone was biologically related to, and with reasonable confidence, roughly where they grew up. But it cannot by itself tell them what social role a person actually occupied, what language they spoke, or how their community understood their identity. The debate over how to interpret the Birka grave has not fully closed, and researchers on both sides of that argument continue to publish work refining their positions. A reminder that a single genetic seat result, however clear on its own technical terms, rarely settles every question a discovery like this raises. Similarly, the broader genomic surveys of the Viking world have left open questions of their own. Why certain regions of inland Scandinavia remained so genetically stable, while coastal trading hubs diversified rapidly. Precisely how cultural identity and genetic ancestry interacted at the level of individual communities and households, and how many more graves scattered across the Viking world, if subjected to the same kind of direct genetic testing that resolved the mystery of BJ 581, might turn out to tell equally unexpected stories. There is also the broader question of scale. Even 442 genomes, an enormous number by the standards of ancient DNA research only a decade ago, represents a tiny fraction of the total population that lived, traded, and moved across the Viking world during those 3 centuries. Every additional grave that gets sequenced has the potential to either reinforce the patterns already documented or complicate them further. And researchers in the field have been candid that the current picture, however much more accurate it is than the one it replaced, should still be treated as a work in progress rather than a finished map. What the last decade of ancient DNA research into the Viking world has established beyond serious dispute is that the popular image of the Viking, inherited largely from 19th century Romantic nationalism and 20th century popular culture, was never a particularly accurate reflection of the genetic and social reality documented in the graves themselves. The people who lived, traded, raided, and settled across that world were more genetically diverse, more mobile, and organized around more flexible notions of identity and status than the stereotype ever allowed. And in at least one very well-furnished grave outside Birka, buried more than 1,000 years ago with a sword, a spear, 2 shields, and 2 horses at her side, the evidence now says plainly that at least one of history's Viking warriors was a woman all along.